Hello?
Hello. I guess my microphone is on.
Yeah. How are you?
Well, I got my model to work.
Good.
At least one of the hexagons to work. So it's easy to make a 3 or 7 hexagon model from that because you just copy it.
Okay.
So maybe by this Friday I will have something, or Thursday I guess.
Yeah.
Is when I need it for. I ran into something interesting here just now. It says, cell mechanics dissecting the physical responses of cells to force, and it is a 2009 article,
Wow.
so I didn't know about this.
Okay.
So I guess I got to read that.
Yeah.
And then there's one called Computation of the Effective Mechanical Response of Biological Networks, Accounting for Large Configuration Changes. So that's a good one.
Yeah.
Do you want the reference?
Yeah, put them in the chat.
Okay, because this one on biological networks, I'm going to have to look at it closely, and it includes some lovely math, of course.
Oh, yeah.
Anyways.
Yeah. Put those in the chat. Actually, the first thing that I wanted to mention was that we had the annual meeting for OpenWorm this past week. So that was pretty good. Actually, we had a lot of the people who are regular senior contributors there. We have a board of directors who's also there, and I presented on some of the things we've been doing in DevoWorm, and they were kind of excited. They were always excited to hear about it. So I don't know if they're gonna if there's any sort of collateral benefit of that. We'll see. But so that was successful. I also did a presentation on the YouTube channel that kind of went in more detail. I only had like 5 minutes at the meeting, but I had 30 minutes on the video, so that's up on the YouTube channel. And then I'm going to be getting out a video on DevoLearn and like a longer form sort of state of the project where we are on that. Okay. So it should be good. Hello, Dick. How are you? Hi. So, yeah, there are your two references. So the first is from 2015, Computation of the Effective Mechanical Response of Biological Networks, Accounting for Large Configurational Changes. So this is like like geometric displacements or mechanical displacements.
Yeah, it's I haven't looked at it too much, but it certainly is along the lines of what I have to look at. So,
oh yeah,
yeah, that's good. Shows a deformed beam, and yeah, only it's looking at a red blood cell, which is not something I wanted to get into.
Yeah.
Yeah, there's a red blood cell and something else. Got something, a 4-fold connectivity and a 6-fold connectivity. And so, intermediate filament network, and it's a lattice, square lattice. No, they don't go like that. Well.
So they have this 4-fold and 6-fold. Does that mean that it's just connected to 4 other things or 6 other things? Is it like those?
Yeah, the 4-fold is just the square lattice, and the 6-fold is a triangular type lattice.
Oh, okay.
And don't know if you can see that. Oh, okay, not good.
Yeah, okay, I see it now. 1, 2, 3, 4, 5, 6. Yeah.
Yeah,
So it's 6 coming together.
Yeah, 6 is. with triangles in it.
Yeah.
Which is more, that would work better with the finite element analysis situation, because that's what it always does to your drawing.
So this is the first paper. Susan showed the picture of the this fourfold and sixfold connectivity in mechanical networks. So that's where you either have 4 squares that come together at a point, or 6 triangles that come together at some point. And this is in this first paper, Computational of the Effective Mechanical Response of Biological Networks Accounting for Large Configurational Changes. And then...
Okay, comment on that. I think Wayne Broadland did a paper a long time ago on those forces at those junctions. I don't recall the name of the paper. Let me try to look it up.
All right. Yeah. And then the second, so that was the first paper. And when I saw large configurational changes, I wondered 2 things. One, are they kind of moving towards entropy, I guess, or configurational entropy? And then second, you're talking about large changes to networks. So, what are the conditions under which those changes occur? Or what is that? That's some sort of dynamical modeling. And Susan was talking about how, we might use...
I'm showing Susan's mute button on.
Oh, no. Yeah, it looks like you're muted, Susan. And so, and then the other thing is that, Susan was talking about finite element analysis and some of the things she's explored with that. But yeah, it looks like an interesting paper. The other one is Cell Mechanics, Dissecting the Physical Responses of Cells to Force. And that's Biomedical Engineering Annual Review from 2009. So that's, again, I guess that has a lot of interesting things in it. I don't know. I haven't read the paper, so I don't know. Okay. So that's great. Thank you, Susan, for those. And so, you're working on your paper that you're going to present soon in public. Good luck on that. Okay. Oh, yeah, you're muted. I can't hear you. You want to type it in the chat, that's fine. But all right. Let me share my screen. I have some things, other papers to go through. So, so there's been some interesting work. All right, let's see what Susan. Oh yeah, there's Wayne Broadland's papers in epithelia. Oh, the system's muting you. Yeah, I don't know why. But we have a bunch of citations here on epithelial cells, yeah. So these are all like biophysics. Some other things related to embryo movements and things like that. That's pretty good. I don't know
which paper it was. There's so many.
Yeah. Yeah. Right. Yeah, thanks for that. So the first thing I wanted to mention is that there was there's been a lot of interesting work on nuclear modeling, and some things that have come out recently where people are modeling the nucleus. So this paper here is it's a sort of a prize essay from a graduate student. This is a prize. I think it's Science journal. at Science, and they had like a contest for people to write an essay about their work. So this is a prize-winning essay on this work here. This is imaging nuclear architecture in single cells. Yodai Takei is his name, and so this is using multiplexed imaging, and so this is done to uncover precise three-dimensional maps of single nuclei. So this is his PhD work. I don't know if it's been published or not, but it's pretty close. So the smallest functional unit in our bodies, the cell, is spatially organized with various molecules, including DNA, RNA, and proteins. Studying the subcellular organization of molecules in individual cells is fundamental for understanding diverse cell types and states, from neurons to cancer cells. So, we want to know something about how the cells are organized within the cell body itself. And so the cell's nucleus, a highly organized membrane-bound organelle that contains genomic DNA in eukaryotes, is closely linked with complex gene regulation. Reviewing its three-dimensional organization is of considerable interest to a wide range of fields, including developmental biology, human disease, neuroscience. And so the first sort of nuclear structures were sort of observed by Ramon Cajal using light microscopy in 1910, and now we have modern genomics and imaging technologies. So we still don't really have good direct measurements of the spatial organization. We have a lot of good genomic data. We have a lot of good, we have microscope slices, but we don't have a lot of the 3D architecture, or at least it's hard to get really precise measurements of the 3D architecture. And then, of course, in this group, we talk about 4D architecture, which is plus time. So, a lot of these things are sort of, time is implicit in them. So, we can think of time as a thing there too. But what they're interested in is building like a 3D model almost, or data that would allow you to populate a 3D model of the nuclear architecture. And so, and then this, this allows you to look at gene regulation, the whole process of gene regulation. It's not just like, the cartoons that we have in our textbooks where we have something binding to something else. There's a three-dimensional structure to that. And some of that three-dimensional structure regulates what happens. So this is kind of where it gets into how they're doing this imaging. And so, it's a pretty big challenge. So the ability to connect 3D chromosome organization and transcriptomic information within individual cells would greatly advance our understanding of gene regulation. So to this end, he co-developed intron sequential fluorescence in situ hybridization, or what they call seq-fish. So this is fluorescent in situ hybridization, but a certain type of it. This allows you to use microscopes to profile transcripts at the transcriptome scale, so you can look at transcripts within the cell. seq-fish works by constructing temporal barcodes on single RNA molecules through sequential rounds of hybridization and imaging fluorescently labeled probes. And then the nascent transcripts appear near their genomic loci. We can profile transcriptional activities and chromosome organization in each cell. So it's taking advantage of how this process works spatially, and being able to get the right imaging technology to work, and then getting it's imaging data and translating that into some sort of a model. So they worked on this method seq-fish. They were able to scale up this kind of profiling to the transcriptome scale, so looking at multiple genes and multiple transcripts. And this opened the door to discovery-driven single-cell spatial transcriptomics. So this is where we have a single cell, we can look at the spatial distribution of transcripts. And so this is, they were working with mouse embryonic stem cells, and in those cases, nascent RNA synthesis tends to occur at the surfaces of individual chromosome territories, whereas relative positions of chromosomes in the nucleus are variable. So this is where you have this RNA synthesis occurring near the chromosomes. So this is within the nucleus. And then the relative positions of chromosomes in the nucleus can be in different places. So you basically can ID the positions of chromosomes by these signals that are associated with nascent RNA synthesis. And so these kind of transcriptome profiles accurately capture differences in cell states. So interestingly, if you have pluripotent states or a cell within cell cycle, you can do different interventions to sort of synchronize cell cycle or identify different periods of cell cycle or identify the pluripotency state of the cell. So they worked in mouse embryonic stem cells, but you can also work in other types of cells and different states of differentiation to get different, profiles. So then, you want to understand the nuclear architecture, you need to record the organization of chromosomes, including non-coding regions, and then simultaneously compare them with higher order spatial relationships between chromosome structures, nuclear bodies, chromatin states, and transcriptional states within a cell. And so this is where they developed this spatial genomics approach. So this is some of the figures here. This is where they did this method seqfish. And so they're looking in a single cell. They're looking inside the nucleus. So this is 5 microns of scale bar. So we're dealing with a pretty small system. And we do sequential rounds of imaging, and we get these profiles where we have these marks, in the image. And those are just the fish profile. And so you can see that pattern here. The idea is that the signal will correspond to the chromatin wherever it is in the nucleus, and so you can reconstruct things like that. And then this is a sort of a false color model of some of this. So this is the nucleus in a mouse embryonic stem cell. This is actually the nucleolus. So we're dealing now with different. So this is the nucleolus of the mouse embryonic stem cell. You have 20 chromosomes, 15 subnuclear structures. This is actually down here. This is the nucleus. I'm sorry. So this is the nucleus of the mouse embryonic stem cell. This is chromosome 1. So they've been able to localize chromosome 1 and chromosome 3. So you can see that you have this. They know from like a genome sequence where that chromosome is in the genome sequence, and they can localize chromosome 1 because they can take they know the transcriptomics, they know where some of these things are, so they can map out different chromosomes. Chromosome 1 is here, chromosome 3 is in these regions. And so you have this, this is what they're getting. So then this is the nucleolus stain, this is the RNA polymerase 2 stain, these are the nuclear zones. So this is actually where you have these structures within the nucleus. You can see here you have these blue areas. These are nucleolus. The RNA polymerase 2, which is the part of the transcriptional machinery. You have these spots here. It's hard to see in this merged image, but down here you can see it. And then nuclear zones are here. And then this is looking at diverse cell types in the mouse brain. So this was in stem cells. So they were looking in the stem cells. But then if you look at different types of cells, you can see that if it's an inhibitory neuron, an excitatory neuron, or an astrocyte, the nucleus is structured differently. So in the inhibitory neuron, you have this, I guess this is nucleolus maybe, and it's in different places. The RNA polymerase is in different places. And so the implication is that it's poised in different transcriptional states, perhaps. I don't know exactly, but it's definitely suggestive that there's a different transcriptional profile, which we already knew, but I mean, this is kind of confirming kind of what that looks like. So this is a first pass at this, I guess, of modeling, and so it's really interesting to see because there are a lot of questions you might raise from this. So it's definitely something that you can merge with other types of spatial transcriptomics to get interesting answers to questions. And so we're at the very beginning of this imaging-based single-cell multi-omics era. I anticipate an explosion of imaging-based single-cell multi-omics in the coming years, in combination with advanced computational and modeling approaches that will accelerate the discoveries of fundamental principles in single-cell biology and beyond. So that's that kind of work. So that's one example. And then this is this is the mechanobiology of nuclear phase separation. So this is a review article from 2022. And, in this paper, they're talking about the nucleus, and they're looking at the mechanobiology of the nucleus. So in the last paper, they talked about the transcriptomics of the nucleus and kind of profiling that. In this paper, they're talking about the mechanobiology. So the cell nucleus can be thought of as a complex, dynamic, living material. So we think about the cell, but we don't, the nucleus is also this kind of thing, which functions to organize and protect the genome and coordinate gene expression. So again, the nucleus is coordinating gene expression, it's protecting the genome, and it's organizing the genome as well. These functions are achieved by intricate mechanical and biomechanical interactions among its myriad components, including nuclear lamina, nuclear bodies, and the chromatin itself. And so while the biophysical organization of the nuclear lamina and chromatin have been thoroughly studied, the concept that liquid-liquid phase separation and related phase transitions play a role in establishing nuclear structure. This has been sort of emerging recently as a thing to think about. We think a lot about the biophysical organization of chromatin, and that's been studied in the nuclear lamina, but then there are these things like liquid-liquid phase separation and other kinds of phase transitions. And you can see in the data in the previous paper that you have these different sort of profiles, spatial profiles for different things going on in transcription. So, you have different spatial locations of things, different spatial locations of chromosomal products and things like that. So these things change, of course, when the cell state changes. And so if that's the case, then there must be something else going on there. There must be dynamics that go from one cell type to another. And so what do those look like? And obviously, we maybe kind of know why. It may be obvious, but how that happens is not really clear. And so these phase transitions may be very important. Phase transitions are likely to be intimately coupled to the mechanobiology of structural elements in the nucleus, but their interplay with one another is still not understood. Here we review recent developments on the role of phase separation and mechanics in nuclear organization, and discuss functional implications in cell physiology and disease states. So this is definitely, kind of a little bit different take on it, but I think it's useful because it has a lot of sort of gives us sort of another perspective to the other papers. So, we're talking about the nucleus, how it regulates gene expression, but then also what kinds of structural aspects of the nucleus are important, not only for that, but for differentiation as a whole. So this, shows sort of these phase separation mechanics in nuclear organization. So we have these multi-phase structures here. We have viscoelastic environment. So we, have something that is of that physical property. It restricts condensate growth. So you don't have condensates. You have this area where things can move around at different places in the nucleus. You have the nuclear lamina, which is the sort of the outside boundary. And there you have lamins and chromatin, nuclear bodies inside. The lamins hold it in. And so this is, a porous structure, but also it has this structure on the outside that serves as a boundary. And of course, as things constantly move around, it needs to be flexible and move with those things. And then this is euchromatin and heterochromatin. It's sort of like this web inside. And so this is showing examples of this where the heterochromatin is more densely packed than the euchromatin. And then these are regions of the coordinated chromatin movement. So there's some synchronization of chromatin in the nucleus as it's moving around. So yeah, they get into some of the mechanics. They do some microrheology. They're doing some different things with the nucleus. They're able to use micropipette manipulations, aspiration, and then atomic force microscopy, where they apply a force to the nucleus and they measure. So this is a force versus extension graph. You have strain stiffening, you have the spring constant, and then you have strain softening. So you have these different physical properties of the nucleus. If you perturb the nucleus in some way, you can induce forces that kind of reveal the properties of the material here. So that's something that's been done. This is just showing the single particle tracking that you can use here. So they have subdiffusive, diffusive, and superdiffusive behaviors all in the nucleus. So this is where you can get a handle on the viscosity of things by looking at how they diffuse through the nucleus. So subdiffusive just means that it's very, maybe there's a very viscous fluid. Diffusive is maybe neutral, and superdiffusive is less viscous. And so this just shows on a log-log scale what that looks like. Okay, so we have some things in the chat here. Any mention of chromal linkers? I didn't see that in either of the papers. Are nuclei the two cells of the same kind similar? That could be, maybe not. We don't know. I guess I don't know how many they've observed. Like I don't think it's been well characterized for a cell type. So if you have a stem cell, it might look very different from like some stem cell type to stem cell type. So if it's an embryonic stem cell or a bone stem cell, there are different types of stem cells that we can look at. It's, I think this is a kind of like a first pass at it. So, and then, of course, they've observed differences with other cell types, but, I don't know what those differences mean other than that they're different. So, Susan says, strain stiffening, I was told, happens in steel when carbon is incorporated into the iron. Yeah. Yeah, so I guess they quench it. I guess that would be the term, or not. Well, anyways, yeah, they dope it, I guess, with something. So they put something in it, change the properties of the material. And then I have a diagram showing vimentin incorporated into actin in that way. Yeah. Okay, yeah, find the reference. I think Dick is typing something. Okay, so yeah, that's kind of discussing viscoelasticity. Viscoelasticity, it's an important property of the nucleus. But again, we don't know a lot about the absolute dynamics here. It's just kind of people have been exploring this. So let's see. So despite the power of these approaches, passive microrheology generally requires that the material being studied is homogeneous and in equilibrium. So that can be a problem in a lot of cells where you have, like in developmental contexts, where you have things changing. And so, yeah, we can't really look at heterogeneity and out of equilibrium activity. It's not as reliable of a method. So you can do different types of microrheology, but it's hard to implement. So in human cells, it's been shown that chromatin exhibits adenosine triphosphate dependent micron scale dynamic correlations. That's ADP dependent micron scale dynamic correlations. Similarly, chromatin displacement has been shown to be highly heterogeneous, with particle image velocimetry suggesting that the local viscoelasticity of the nucleus varies by compartment. So this is where we get different properties across the nucleus depending on sort of how it's compartmentalized. Despite these confounding aspects of a typical cell nuclei, the Xenopus laevis oocyte provides simpler contrasting examples. So this is in Xenopus. Mature Xenopus oocytes are around 1 millimeter in diameter and relatively homogeneous, featuring a low chromatin volume fraction. So less chromatin, I guess maybe the less chromatin, the more homogeneous, although I don't know if that's true or not. That just kind of seems what may be the case. Analysis of the MSD of beads injected into the oocyte nucleoplasm compared with active microbiological measurements of beads sedimenting through the nucleoplasm under gravity. So they're kind of testing that sort of the viscosity here, and they're making these different microbiological measurements. Suggests that this particular system is not strongly out of equilibrium. So we have to kind of think about how much out of equilibrium a certain nucleus is to sort of get a sense of how accurate our measurements might be. So let's see. In addition to suggesting that the chromatin network is highly viscoelastic, subdiffusive motion of nuclear components has been the subject of much experimental and theoretical study due to the relevance of the first passage time or genomic search problem. So inside the crowded nucleus, critical processes may be limited by the time it takes for two loci to diffuse into proximal locations. So this is where you have this, in the crowded nucleus, things are moving around, and it takes time for things to move to different locations or to diffuse. So I showed those examples of subdiffusive, diffusive, and super diffusive, profiles where it either moves just a tiny bit around its origin and moves kind of in a healthy Brownian motion, or it's really kind of moving in all different locations, very, very diffuse from where it started. That's what they're talking about. That it takes time for that kind of diffusion, but it may be heterogeneous as well. So this kind of, these critical processes may be limited, but these are things that happen. So this is including enhancer-promoter interactions and homology search during homology-directed repair of DNA double-stranded breaks. So the sort of the larger scale changes in the nucleus can be affected by diffusion, and then diffusion itself can be affected by these different types of activities going on in the nucleus. In addition, recent work has suggested that chromatin may lie near a sol-gel transition to facilitate a transition to rapid liquid-like dynamics when functionally required. And that DNA damage may drive directional motion of chromatin. So that's interesting. So this may not be just kind of a stochastic process. It may be driven by certain things going on in the nucleus, certain functions. The presence of liquid-like nuclear condensates may facilitate this genomic search. And we know that although condensates are sort of inhibited here, they can still occur. Enhance transcription by providing an environment with rapid liquid-like dynamics, which have been observed coupled to transcription. That's reference 49. Or excluding non-specific bulk chromatin, that's reference 50. So they've done a lot of kind of review of the literature on this. There's a lot of stuff suggesting that you have these different, maybe specialized mechanisms that occur that, optimize different processes in transcription. So this is interesting. Together, this evidence may suggest that condensates play an important role in spatially organizing chromatin. So I think we've talked about condensates before, so they can actually organize chromatin. They're not everywhere in the nucleus, but they do exist, and they do serve this kind of maybe adaptive function, and regulating the viscoelastic properties of the local environment of the nucleus, although their role in the maintenance of bulk material properties remains unclear. So we don't know how it actually regulates, how like if you were to do some sort of microbiological probe, how that would change the behavior of the nucleus. Would it become more, rigid, or would it become more flexible, or whatever? So, yeah, this kind of talks about, more about this. Yeah, so this kind of talks about phase separation. So you have these different phases in the heterochromatin domain. So this is, here's some nice graphs if you're interested in how they kind of measure this. And yeah, so this is basically the conclusion. Phase separation provides a compelling model for the functional organization of biomolecules in the nucleus and elsewhere within living cells. In recent years, significant progress has been made to move beyond overly simplified and sometimes only qualitative models towards considering the ramifications of the uniquely complex environment of the cell as on phase separation. In particular, the mechanochemical complexity of the intracellular environment gives rise to unexpected behavior and indeed potentially even new physics. So that's interesting. These include behaviors not predicted by the simplest two-phase liquid. So there are different binary models that you can use to look at this. The presence of many stably sized aspherical droplets or a composition-dependent saturation concentration. So there's this idea of one particularly intriguing intracellular complication is the viscoelasticity of the cell. So viscoelasticity is probably a key driver of a lot of these processes, but we don't know, we can measure it, but we don't know what it means or what sort of the rules are of the nucleus. While both nuclear bodies and nuclear mechanics have been separately investigated for decades, conceptual framework of nuclear bodies as phase-separated condensates provide an opening for understanding their interplay with nuclear mechanics. So this idea of phase-separated condensates and what nuclear bodies, how their dynamics within the nucleus, that's something we don't, that's maybe a new area that might emerge from this work. The effects of the mechanics of the environment on phase-separated droplets has only recently begun to be appreciated in biology. But we need more work and methods to measure the physical parameters such as surface tension, permeation stress, and elasticity. And this theory predicts this will govern the condensate behavior in cells. And so then the other complication and maybe opportunity is to understand non-equilibrium activity in an intracellular context. So what is the sort of the equilibrium activity within the cell for different cell types? And we don't really know how to deal with that. Linking mechanics and phase separation, when we approach this with our current approaches, it assumes thermodynamic equilibrium, but of course we can't assume that in a biological system. There's a lot of biological regulation going on, so it's in a high state of non-equilibrium. And so we can ask some questions about this, but to get the answers, we need to have sort of non-equilibrium friendly methods. So some of these questions are: do cells use condensates as sensors of mechanical stimuli? So, for example, to alter the epigenetic state and stiffness of the nucleus in response to external forces. So, there's stuff going on between the nucleus and the rest of the cell. And are those regulated by external forces? Are there, like there's stimuli that activate first messenger genes and things like that? Are those things regulated by some sort of physical mechanism? Can condensates also directly alter local nuclear viscoelasticity to respond to changing mechanical requirements? So is viscoelasticity the regulating factor are we getting this viscoelastic regulation in response to some of the things that are going on in the cell's mechanical environment? And so condensates may play a role for this in different ways at multiple time and length scales. This could range from maintaining heterochromatin-mediated bulk nuclear mechanics to coordinating protection and repair in response to DNA damage. So if this is the case, then we have a lot of maybe really interesting answers to these questions that go beyond this immediate model of the nucleus. Yeah. So this is a nice review paper. I wanted to go over that because we haven't really given the nucleus its due, I guess. So let's take a look at the chat and see what we have here. So yeah, this is where we were. We talked about this. Work by R. Nagel showed that chromosomes have circular ordering. And this is in eukaryotes, so this isn't like a circular chromosome. This is actually just the circular ordering of the chromosomes. So Susan's has, I guess these are the references that she put in. The first is non-channel mechanosensors working at focal adhesion stress fiber complex. So this is one article, and then the other article is mechanics and functional consequences of nuclear deformations. Yeah. And then Nick says, or an intellectual trap. So that's, yeah, it probably could be. I mean, it's not an easy area to study. And then Susan says the mechanics of cells is affected by pH. I can share my screen to show the vinculin-actin connection. Yeah, would you like to share your screen? Okay. All right, there we go. There we go. Yeah, so that's the intermediate filament vinculin associated with the actin. So it looks like there's a flow, and yeah, and the vinculin is being added in after the force, after the retrograde flow. The actomyosin force is going in one direction, and then the vinculin is being added in, and yeah. So this is non-channel mechanosensors working at focal adhesion stress fiber complex. This is from this paper, right? Okay. That's great. Thank you. All right. Oh, I hear something. Is that just the microphone? I don't. Okay. Yeah, I don't know. I don't know why everything is strain softening or strain hardening. What does that mean? Oh. The vinculin incorporated with the actin, yeah. So it depends on the softening or hardening, or people don't know what it is, or so yeah, they don't know. Yeah. So it's somehow being incorporated in the actin, and they don't necessarily know why it's happening, but it's there. The definition of strain hardening was steel, so that was the example of steel that was strain hardening. Yeah. Well, that's a nice find. Yeah, thank you for that. It's like Dick is typing something in. The circular ordering is consistent with Maniotis's observations of DNA chromal linkers. Okay. Might be worth reviewing. Yeah. If you could send me the reference for that, I could do a review on it or look at it in the meeting, because it might be a nice. Because there's probably a lot of literature on the nucleus that, and they just reviewed a little bit here in this paper, or in these two papers, but. Okay. So I don't know why our audio is the way it is today, but hopefully we do this again. It won't be like this. But I don't know. It was like we were playing like audio tag or something. I don't know. So yeah, thanks for that. So that's good. I think we'll end for today. So you wanted to mention anything else? Dick might have something he's typing in. Nigel's papers. Okay. There we go. There's Nigel, Velasco, Anderson, et al. Telomere associations in interphase nuclei, possible role in maintenance of interphase chromosome topology. And that was from 2001. Okay. Yeah, it's interesting. Might have to review that maybe next week. Follow up on it. A couple of things I'd like to follow up on, actually. But. All right. Well, thanks for that. So, all right. Well, thanks for bearing with me on the audio. And, all right. Have a good week. Thank you. Bye. All right. So now I'd like to cover a few papers, two of which Susan brought up in the meeting in an area we're calling morphogenetic physics. So the first paper Susan talked about was this paper on computation of the effective mechanical response of biological networks, accounting for large configuration changes. So this is the one that was intriguing from the standpoint of configurational change and these responding biological networks. So let's read the abstract. The asymptotic homogenization technique is involved to derive the effective elastic response of biological membranes, viewed as repetitive beam networks. And we'll see what those look like later. Thereby, a systematic methodology is established, allowing the prediction of the overall mechanical properties of biological membranes in the nonlinear regime. So this is an important concept that we have biological membranes, we're modeling them with mechanical models, and we're modeling it so that we can capture this nonlinear regime. This is if you force the membrane, it has a response that is not linear, reflecting the influence of the geometrical and mechanical microparameters of the network structure on the overall response of the equivalent continuum. So this is just talking about the sort of the network structure itself and the responses. Biomembranes networks are classified based on nodal connectivity, so that we analyze in the work 3, 4, and 6 connectivity networks. And this is referring to like fold connectivity or fold, I don't want to say symmetry, but it's similar to that, which are representative of most biological networks. The individual filaments of the network are described as undulated beams prone to entropic elasticity, with tensile moduli determined from their persistence length. The effective micropolar continuum, evaluated as a continuum substrate of the biological network, has a kinematics, which means it has a sort of a motion dynamics, reflecting the discrete network deformation modes, involving a nodal displacement and a microrotation. The peptidoglycan network may exhibit a reentrant hexagonal configuration due to thermal or pressure fluctuations. So this is translating back to the biology. Our mechanical network that we're simulating is actually a peptidoglycan network, and that's the sort of the surface of the cell. We're taking the properties of these macromolecules and we're sort of fitting it into this model. So they say this sort of network may exhibit a reentrant hexagonal configuration due to thermal or pressure fluctuations, for which micropolar effects become important. The homogenized responses are in good agreement with finite element simulations performed over the whole network. The predictive nature of the employed homogenization technique, as well as the identification of a strain energy density of a hyperelastic model, for the purpose of performing structural calculations of the shape evolutions of biomembranes. So this is their model here. We have what they call 3-, 4-, and 6-fold connectivity. So they're taking a look at these macromolecule networks. We have sugar rings linked by peptides, and this gives us a 3-fold connectivity. So this 3-fold connectivity has junctions where you have 3 elements coming together at one point. So this is an example here. Let me draw this out on a board. So a 3-fold connectivity is something like this. So we have 1, 2, and 3. So there are 3 spaces in between this junction. So this junction is formed by some vertex in the network. So that vertex is this 3-fold complexity. Okay, so you have, and there are, of course, mechanical advantages to this, and there's structural advantages to this as well. So let's go back to the example here of 4-fold connectivity. Actually, here's something from the nucleus. And so we have a nuclear membrane, we have pores in the nuclear membrane, we have the nuclear lamina inside the nuclear membrane, and then the area inside the lumen-containing DNA. And so this is what we call 4-fold connectivity, which is this nuclear lamina. So you can see the lamina, which is this square pattern, and so it gives us 4-fold connectivity, and you can see that there are 4 elements coming together in one spot. So this gives us something that looks like this. We have a vertex here. And again, we have 1, 2, 3, and 4. And so this again has different mechanical and structural advantages. So this is 4-fold connectivity. And so then let's go back to the 6-fold connectivity example, where we have, this is from a red blood cell. So this is what Susan mentioned in the meeting. You have these irregular shaped edges. So these are erythrocyte network. And so again, our 6-fold connectivity, this is the vertex. And we have 6 elements. Like so. So again, structural and functional advantages to that. So you see all these different types of connectivity and different types of tissues and structures in structural biology. And then the question is, why do we have these different types of connectivity? Is it that it's optimizing for the different conditions that are required for that thing? So for example, red blood cells, erythrocyte networks are optimized for cells through the bloodstream. The nuclear lamina is optimized for a structure that has gives some rigidity to the nucleus, or this set of sugar rings and peptides. We know that hexagons give really optimal packing structure, so maybe there's something about that, or maybe some sort of structural stability. So we don't really know why that they are the way they are. It's just that we can identify these different types of connectivity in these networks. So this is again an example of peptides and sugar rings, this peptidoglycan network, and we can model this in terms of nodes and edges. So let's read a little bit about 3-fold connectivity. So we have, it is believed that this molecular and isotropic organization is dictated by design principles of the cell, such that the stiffer chains act as reinforcement in the direction that sustain the maximum stress. Knowledge of the mechanical properties of peptidoglycan is of importance for understanding bacterial growth and form. And the angle is used as a descriptor of the topology of the glycan network. So we're looking at the angle radiating from those nodes. The vertex is sort of the organizing point in different parts of the network. This varies in between 5 degrees and 20 degrees according to the dimensions of the molecular chains of the unit cell shown in figure 2. We evaluate the average Young moduli of glycan at low temperature. The Young's modulus are elaborated from the persistence length according to this relation. Now let's look at fourfold connectivity. So the nuclear lamina contains junctions that have fourfold coordination. The nucleus is bounded by 2 membranes, and the lamina lies in the interior of the nucleus, adjacent to the inner nuclear membranes. So this is a biological square network. The animal cytoskeletons consist of 3 interconnected filament systems: the actin-containing microfilaments, microtubules, and the lesser-known intermediate filaments. Intermediate filaments are known to impart mechanical stability. Single intermediate filaments can be described as a flexible, extensible, and tough material, which has important implications for our understanding of cell and tissue mechanics. So the nuclear lamina is composed of these. It's a meshwork at the nuclear membrane that protects the genetic material and regulates gene expression. The geometry of the meshwork is obtained from experimental observations of the nuclear lamina. They use Xenopus as a model organism. The network is composed of 2 sets of neuro-orthogonal intermediate filaments with a lattice constant of 50 nanometers and a diameter of 10 nanometers. Let's talk about 6-fold connectivity. So the membrane of the erythrocyte has both a low density and high flexibility. Furthermore, the corresponding lattice is 6-fold connectivity, with however 3% and 8% of the junctions presenting 5 or 7 folds respectively. So this isn't uniform across the erythrocyte network. You can have 6-fold connectivity, but you can also have 5 and 7-fold connectivity at low frequencies. So this is interesting because this isn't a uniform network. This is an anisotropic network with respect to these sort of junctions. And so why would we have this difference in junctions, given that we don't necessarily see this in the other networks? So then they just kind of assume a rectangular beam section and they simulate that. They don't really give an answer as to why that is. Okay, so some of the things we can do with this kind of a network, and in this case we're looking at a 4 connectivity network. So we're looking at something that you might see in the nuclear lamina. This is the topology of the 4 connectivity network here, and then the nonlinear bending response. So you can look at this network and the vertices that form the junctions, and then you can take that area, the area between the vertices especially, and get a nonlinear bending response for that local part of the network. And so this is where we have a linear and a nonlinear response. So this is the Young's modulus, and then this is the length measurement. So we can see as this thing is deformed, we have different properties. Okay, the second paper Susan talked about in the meeting is this one here, Cell Mechanics Dissecting the Physical Responses of Cells to Force. So this is a review article. This is from 2009 by Brenton Hoffman and John Crocker. Let's read the abstract. It is now widely appreciated that normal tissue morphology and function rely upon cells' ability to sense and generate forces appropriate to their correct tissue context. Although the effects of forces on cells have been studied for decades, our understanding of how these forces propagate through and act on different cell substructures remain at an early stage. So they're leading up from the last paper, you can identify these different types of networks, and we can see that they behave differently. And so the consequence of that is that if you apply a force to a biological tissue, if it's made up of these different type of network topologies, you end up with different types of effects. The past decade has seen a resurgence of interest with a wide variety of different micromechanical methods in current use that probe cells' dynamic deformation in response to a time-varying force. So, we have this variability based on the physics, but we can also have a time-varying force from the outside. The ability of researchers to carefully measure the mechanical properties of cells, subjugated to a wide variety of pharmacological and genetic interventions, however, currently outstrips our ability to quantitatively interpret the data in many cases. Now, despite these challenges, the stage is now set for the development of detailed models for cell deformability, motility, and mechanosensing, and we can look at that at the molecular level. And so this is a very long review article. They talk about seeking a consensus description. So a lot of the papers we're doing in the meetings are sort of recent papers. They're from the last several years. But this review is from 2009, so they're talking about how you might engage in this activity. And so they're providing this ways of getting to a consensus description. So, we can measure things over a wide frequency range. We can account for variables by probing connectivity and cell heterogeneity, which we saw in the last paper. We have the effects of non-thermal random forces. We have evidence for mechanically distinct structures, so we just saw that in the last paper as well. And we need a consensus description for unperturbed cells, or an equilibrium state. And so these are all things that people have worked on, and people are working on the details of this. The second part is lack of consensus. Actin and myosin perturbations and their interpretations. So they talk about actin perturbations, where you perturb actin molecules. You have vasoactin compounds and myosin-specific perturbations. You have universal versus non-universal exponent approaches, and a modified consensus description, adding a distinct actin myosin network. And then the mathematical properties of power laws can confound interpretation. So these two are kind of interesting because these are 3.3 and 3.5, because this is sort of the meat and potatoes of complexity theory and sort of this concept of universality. In other words, we expect things to behave in the same way across systems. And we can use, single tools to get at different questions. And the question is this something that we should be doing in this kind of environment? So, there have been papers that have criticized the application of power laws in physics and in complexity theory. But there have been a lot of papers that have supported power law interpretations. So power laws are, somewhat controversial maybe. Are they applicable across domains? What do we make of a power law exponent? These sorts of things. And then, we have these universal versus non-universal exponent approaches. So we usually calculate some sort of exponent on our data. In the last example, we had a linear versus a nonlinear function. And you would take the exponents from those, at least the nonlinear function, and you would compare it with different types of systems. And the question is that something that you can do? Is that something that's reliable? Does that bring us towards a consensus between cell types? And then having a model of the cell mechanical response. So there are different models that people have advocated for. So there's this, they have an overview of the model space, which includes all of the models that, the main models and some of the more obscure models. Some of the main models include the salt gel hypothesis, a tensegrity model, the soft glassy rheology model, active gels and dynamic crosslinks, and other emerging models. This is something to follow up on. We, of course, are supporters of the tensegrity model in this group. But there are other models, active gels, soft glassy rheology model, which I've kind of touched on as well. This is something, again, we need to sort of get a sense of what kind of model is best, or what kinds of models are best for describing these sorts of things. And then the implications for cell biology, we can talk about mechanotransduction, which is a lot of what we're sort of our immediate interest here with Susan's project, but then other biological processes as well. So I'd like to conclude with going over the abstract. Pretty diverse, but I think they give an idea of how to apply some of these ideas to larger scale systems. So the first paper is from Hernandez-Lopez et al. And this is two fluid dynamics in micron-thin boundary layers shape cytoplasmic flows in early Drosophila embryos. So this is fluid dynamics in boundary layers in embryogenesis. Actually, I read the significance section and then we'll read the abstract. So the significance section reads, embryos and cells often feature substantial flows of their cytoplasmic components. So this is where we have flows, and we've seen this in within the cell, but also within embryos. While their Reynolds numbers are moderate, flows typically involve non-trivial couplings between mechanics and biochemistry. So in other words, if the Reynolds number is really small or really large, we use that as those sort of outliers as an example of a system that's interesting. If a Reynolds number is moderate, we don't really worry about laminar behavior or turbulent behavior. It's just kind of a normal sort of situation. But these flows actually involve non-trivial couplings between mechanics and biochemistry, meaning that the mechanics are interesting even if the Reynolds numbers are within a normal range. Here we couple the mechanics of cytoplasmic flows to the cell cycles of embryonic nuclei and Drosophila embryos. So this is where we have these embryonic nuclei before cellularization in the Drosophila embryo, and we have these mechanics of these flows of the cytoplasm. We show the fundamental and empirical reasons to impose a multi-phase model with both a passive, the cytosol, and an active fluid, the actomyosin network. So we have two components of this model. This is a multi-phase model, so it has multiple components. We have a passive component, which is the cytosol, and an active fluid, which is the actomyosin network. So we have two parts. Entrainment of the passive by the active fluid. So that means that active fluid sort of entrains the frequency domain of the passive element of the model. Dramatically concentrates in a thin boundary layer at the embryo cortex. The model recapitulates experimental flow patterns, explains previously elusive observations, and makes a series of predictions, which is what a good model in theory should do. In particular, the predicted relations between the flow structure and embryo geometry are experimentally confirmed. So what they've been able to do is take a multi-phase model with two components, show how those things synchronize and behave together, and then use it to explain different aspects of the system. So the abstract reads, cytoplasmic flows are widely emerging as a key functional player in development. In early Drosophila embryos, flows drive the spreading of the nuclei across the embryo. So this is where the nuclei spread out across the embryo. You have this flow. They have to be entrained so that they're in sort of a decent position for later development. Here we combine hydrodynamic modeling with quantitative imaging. So they're doing the modeling, but they're also doing imaging to develop a two-fluid model. And this is this multi-phase model that features an active myosin gel and a passive viscous cytosol. Gel contractility is controlled by the cell cycle oscillator, the two fluids being coupled by friction. So this is where we have this, the gel contractility is controlled by some sort of model of an oscillator, and the fluids being coupled by friction, which is a force. In addition to recapitulating experimental flow patterns, our models explain observations that remain elusive and make a series of predictions. First, the model captures the vorticity of cytosolic flows, which highlights deviations from Stokes flows that were observed experimentally but remain unexplained. Second, the model reveals strong differences in the gel and cytosol motion. In particular, a micron-sized boundary layer is predicted close to the cortex, where the gel slides tangentially, while the cytosolic flow cannot slip. Third, the model unveils a mechanism that stabilizes the spreading of nuclei with respect to perturbations of their initial positions. The self-correcting mechanism is argued to be functionally important for proper nuclear spreading. Fourth, we use our model to analyze the effects of flows in the transport of the morphogen bicoid and the establishment of its gradients. Finally, the model predicts that the flow strength should be reduced if the shape of the domain is more round, which is experimentally confirmed by mutant phenotypes. Thus, our two-fluid model explains flows and nuclear positioning in early Drosophila while making predictions that suggest novel future experiments. So this figure 1 shows a scheme for the mechanochemical coupling underlying our model for cytoplasmic flows. So this is a sketch depicting a slice of the anterior side of the egg. So this is sort of the front part of the egg, along with the ingredients of our model and their location in space. So you have the nuclei here, and then you have the flows, the sol flows, the gel flows, which go up to the surface, and then this green, which is the meiosis. And then this on the right here, we see captions and interaction sequences detailing the mechanochemical coupling that result in nuclei being transported in the embryo. Meiosin 2 drives gel flows. There's friction that interacts with sol flows. There's advection, which interacts with the nucleus, or in any one nucleus. This activates PP1, which is a factor that promotes meiosin 2. So you can see this is a positive feedback loop containing multiple components. And so this is an example of the embryonic flows. You have the Drosophila embryo. You have these flows that occur. Our second paper is forceful patterning, theoretical principles of mechanochemical pattern formation. So this is Jen Ramboutz, Jenna Elliott, and Anna Ersberger. Biological pattern formation is essential for generating and maintaining spatial structures from the scale of a single cell to tissues and even collections of organisms. Besides biomechanical interactions, there is an important role for mechanical and geometric features in the generation of patterns. We review the theoretical principles underlying different types of mechanochemical pattern formation across spatial scales and levels of biological organization. So they kind of go through more of a theoretical approach to this. And the last paper was very much an empirical paper. This is a theoretical paper. So in the following, we introduce some of the theoretical concepts used to study pattern formation. We consider patterns as regular spatial features that consist of many units. By units, they mean cells or molecules, where the characteristic length scale of the pattern is much larger than the size of a single unit. A pattern can be structural, characterized by a regular shape, or it can be manifest as regular variations in the concentration or density. Typically, a pattern consists of repeated features. In that case, we identify a pattern's length scale with its wavelength, that is, the distance between repeated features. We refer to the length scale of individual units, L, as microscopic, and the pattern length scale as mesoscopic, relative to the macroscopic size L of the whole system. And so then this is basically a local global sort of mapping where you understand pattern formation and how patterns are repeated, and using the patterns as sort of a unit of analysis that is larger than a single unit. So you can look at emergent phenomena, you can look at local versus global effects and things like that. So in some cases, this allows for a statistical description. So when the spatial scales are well separated, the large number of macroscopic units that make up a wavelength of the pattern permits a statistical description. An example such as a description as a concentration field that keeps track of only the average number of units per length, area, or volume at each point in space and time. Separation of spatial scales in many cases also implies a separation of temporal scales, because things may happen like from one end to the embryo to the other or something like that. So indeed, the mean collision time of water molecules within a typical ocean wave, with a period of 10 seconds, is below 10 to the minus 10th seconds. Within the actin cortex of the cell surface, typical timescales of macromolecular interaction are on the order of milliseconds, whereas they lead to cell-level shape dynamics and flows on the minute-to-hour scale. So this is interesting that you have these different timescales that they put into their theoretical considerations as well.